Good morning. Just give you a second to find a seat. Good morning all, and thank you on behalf of ICES for turning out this morning for this second of ICES ASC keynote speeches. We have the privilege today of welcoming a scientist who is a leader in her field, Jenni Prokkola. Welcome, Jenni. Jenni is a senior scientist and research fellow at the Natural Resources Institute in Finland. For the last decade, she has worked extensively across the board in marine and freshwater fisheries, from fishery-induced selection to fisheries behavior to oxygen supply. Jenni is a regular contributor to ICES, and from 2025 will co-chair the working group on science to support conservation, restoration, and management of diadromous species, as well as being an active member of the working group on Baltic salmon and trout. Today, Jenni pursues her passion, understanding the complex life cycles of salmonoid fishes, how they have evolved, and how they cope with human-induced stresses, including climate change, a topic never more pertinent than it is today. And it is this that Jenni is here to talk to us about today. In particular, how genetic variation controls key life history traits in salmon, and how heat waves are shaping salmon populations today. So, please join me now in giving a warm welcome to the fantastic Jenni Prokkola.
Thank you for that amazing introduction. Thanks for everyone for being here bright and early. It is, in fact, my first annual science conference, so it is a great honor to be here and meet everybody at the same time. I don't know many people here yet, but I hope to change that. So I guess most people in this room probably work in some way or other to support sustainable fisheries. We know how important it is that we can predict the abundance of a fish stock and how difficult that is. One of the things I'm excited this week is to learn about the different methods that we have for predicting stock sizes, because I'm relatively new to that field. But in my career, I've mostly focused on within species, among individual variation. And I'll try to convince you how this is important as well. So my main focus is trying to understand how salmon tolerate changes in their environment, also perhaps one day predicting the effects of climate change on salmon populations. But in this talk, I would like you to all take this individual fish perspective with me. Now, the perspective of the individual is that of an ectothermic animal, and that means we cannot overlook the effect of temperature on them. As endotherms, humans probably cannot fully relate to how important it is to ectotherms, how temperature changes their body temperature, but it's nevertheless absolutely central. So the main effect of temperature on fishes is that it increases the rate of biochemical reactions in their body, just because of physics and chemistry. So even in resting state, without any movements, fish need more energy to stay alive when temperature increases. That can be a nice thing, because by increasing food intake, they can increase growth rate and activity when temperatures get higher. By the way, we refer to this basal or resting metabolic rate as the standard metabolic rate in fishes. It's not the full story, because they can't indefinitely increase their food intake and growth rate. And there's a theory that suggests that the limit where this increase is no longer possible is set by their oxygen supply capacity. So usually we are talking about maximum metabolic rate when we mean capacity for oxygen supply. This was already predicted by Frederick Wright like 80 years ago, but it's still relevant today. So he invented this concept of aerobic scope, where the difference between the standard metabolic rate and the maximum metabolic rate determines how much aerobic energy an ectotherm can use for different functions like growth and reproduction. He also predicted that with temperature, this aerobic scope is going to decrease, and that will have severe negative effects on fish physiology. So how well has this been tested in reality and kind of confirmed in reality? So a couple of extensive studies have recently compiled our data up to date and concluded that this fry paradigm matches observed data, usually when temperatures that are tested are higher than what the fish would naturally encounter in their environment. It also matches this more often in adults than in juveniles. So juveniles can sometimes just sustain a high aerobic scope at every temperature. It also matches it more often in marine than freshwater species, probably because marine species have a more stable thermal environment. Another study, which had some really complex analysis I won't go in detail to, but they found that at present climate, present distributions, oxygen supply doesn't seem to be limiting fish distribution, so it means that they should have physiologically enough capacity to deal with every temperature and every condition in their environment, and probably distribution is set by something else. However, when temperatures increase beyond the natural range that these animals have lived in, probably millions of years, then oxygen supply and other systems start to fail. Now, this brings me to climate change, which obviously has already increased this temperature of the planet by more than 1 degree on average. In the best case scenarios, the temperature will continue to increase this century a little bit, but in the worst case scenarios, a lot, and by much longer than this century. Now, this change has only taken 200 years, so species obviously have not had time to evolve in response. And it's not only a challenge for the species, but I think it's a challenge for us who want to understand what is the fish stock size and how much can we fish. Like, without considering this change, we are probably going to fail. And of course, here it is hidden that climate change is not affecting everything equally. It is affecting, for example, the Arctic much more than the lower latitudes, and it is increasing the frequency of extreme weather events. And this brings me to heat waves. And it's a little bit busy graph, but I love showing it. So the IPCC also predicted the frequency of different heat waves with climate change. And for example, with the best case scenario of 1.5 degrees warming, we would still see like a 4-fold increase in heat waves that used to happen once every 10 years, and they would be 2 degrees more severe. And some more rare heat waves, so more severe heat waves that previously occurred once in 50 years, would occur 9 times more often and be 2 degrees more severe. Now, because we are currently not on track for 1.5 degrees at all, we also need to consider the worst options there that seriously make us a little worried. Now, for fishes, the effect of heat waves would depend partly on where the fish lives. Not all aquatic environments will be equally affected. And one environment that is surprisingly sensitive, I guess, is the rivers. This is a relatively big river in Finland, even though it looks like a lake. It's the Tornionjoki, and habitat of biggest salmon in population in Baltic. So rivers experience heat, sunlight, like cumulatively, because the water is flowing during the sunlight, and it can take a long time to reach the sea. Also, they are usually shallow, they can be dark, which further absorbs the heat. So in salmon, this is pretty relevant because they inhabit rivers for half of their life. The juveniles spend several years in rivers, 1 to several years before migrating to sea, and the adults spend summer in this river before spawning. So at both life stages, they can be vulnerable. For example, here is a salmon index river from Finland. It's Simojoki, where I participate in some of the monitoring that we then use for the stock assessment. It's there just south of Arctic Circle. This is an example of a temperature logger we have in the river from last year. So temperatures reach 23 degrees, and several times they go beyond 20 degrees during the summer. And even though it's not particularly hot as such, like there are worse rivers for salmon in other countries, but it's already concerning because salmon growth is predicted to decrease already at 18 degrees, and 23 should be severely stressful. So keeping this in mind, what I want to do as an experimental biologist is test if the aerobic scope of salmon actually decreases as predicted by the fry paradigm. This is still not feasible to do with wild populations. It would be kind of way too difficult to get enough sample size for the sensitive analysis. So we are using hatchery-reared individuals, but they have like an origin in semi-wild and wild genetic background. We rear them in naturally fluctuating water, so the temperature is pretty natural to them. And then during the summer, their first summer, we expose them to this artificial heat wave. So after 1 week at 16, 19, and 22, we measured the aerobic scope of the juveniles. And then because it was cumulative, we also measured the aerobic scope again after the experiment to kind of see that there was no effect of development on that. A few words about the methods if you're not familiar with respirometry, but this is our homemade respirometer that we call SHREC. We measure the maximum metabolic rate of the fish by exhausting them manually. This is important so that they can increase their oxygen supply as much as possible during the recovery. So for the recovery, we put them in the chambers and then measure oxygen consumption during the next few hours, let's say. Then we leave the fish in the chambers overnight so we can measure their standard metabolic rate. At this point, they need to be resting and non-digesting, etc. And well, the results are actually, for once, somewhat expected. We first find that, of course, standard metabolic rate increases with temperature. And here I'm showing you already mass-corrected values. So the value is showing like the deviation from that one predicted based on mass alone. Because, of course, a bigger fish will have higher metabolic rate than a small one, so we're not interested in that. If you look at within 16 degrees, there was no difference in the first and last time point, so at least the difference between temperatures is purely due to temperature. We didn't see anything similar in the maximum metabolic rate, so the fish simply couldn't increase their oxygen supply at the higher temperature, and this means that their aerobic scope decreased, as the fry paradigm already suggested. So, in conclusion from this, the reduced aerobic scope suggests failure to maintain performance at above 20, maybe 22 degrees, in short or long term. It doesn't mean that the fish would immediately die, but it means their performance would deteriorate, and maybe they're more susceptible to disease or growing more poorly. So, to actually start talking about evolution and life history, I'm going to take a little step back. So let's recap salmon life cycle for those of you who don't know. So they spawn in the river, and the eggs and the alevins, which are the larvae, develop in the gravel over winter. Then during the summer, the fry develop in the parr, and the parr can spend multiple years in the river until they're big enough to do the sea migration. Some of the male parr can already reach sexual maturation in the river, and they're called like sneaker male. They will go and try to fertilize the eggs of the really big females. But even after this, they can still smoltify and do the sea migration like the other fish. The sea migration stage is called the smolt, and then at sea, they really begin to grow fast as they change into a piscivorous diet. Now, there's this big variation in how many years the salmon can spend at sea, from 1 to 4 or even 5. And then that will be affecting how big they are when they return to spawn, and how big they are will affect how big is their egg production or fecundity in the river. So this variation is super interesting, and it's also useful for salmon because by being able to flexibly adjust age and maturity, they are a bit more resilient to changes in the sea conditions, and also perhaps more likely to breed with a non-related individual when they come back to their home river and the home territory in the river to spawn. In one study that was done on wild salmon, so genotyping offspring of salmon and assigning it to parents and then parents' sea age, it was found that, yes, even the number of viable offspring increases with sea age. So it's great to be an old and big salmon. You produce lots of offspring, but of course the drawback is you're more likely to die at sea before any sort of reproduction. So overall, diversity is good. There's no one best strategy here. But still, this trait is important, and a few years ago, it was found that it also has a very strong genetic basis. So I will take you through this, if you're not familiar yet. The authors from Finland and Norway, they took salmon scale archives from 57 populations and combined the sea age based on the scale to the genetic variation in the sample. So they used these 200 genetic markers across the genome and associated the markers with the sea age. And here is this Manhattan plot. So that means the whole genome of salmon here is on the x-axis, all the markers they used anyway. So 29 chromosomes, and on the y-axis is the significance of the association. And there was this one humongous peak. So one single nucleotide, which we call SNP, was associated with the sea age. And it actually explained almost 40% of variation. And that was they named it VGLL3 because it was located in this chain that is homologous to a VGLL3 in mammals. And that's how usually fish genes are named. So what it means is that when VGLL3 in salmon occurs as this early maturation version, so allele, when salmon have an early maturation version, they are on average maturing 1.3 years earlier than the late maturation version. Good. And this is useful because now we can just use genomic prediction of sea age and do our studies before any fish mature. We don't have to wait 5 years for them to mature. We can study them based on this genetic trait. Here are a couple of colleagues holding their fish catch. Of course, early maturing ones are very small compared to late maturing ones. So in Common Garden, then lots of projects started to confirm this result, mostly first in Craig Primer's group, and they find that this also affects the male parr maturation. So you remember the male juveniles, so par, can reach sexual maturation quite quickly without going to sea. And it's always related to body size, so generally individuals can't mature without being big enough. But also having this E allele, so EE genotype, it's inherited E alleles from both parents. Those fish are the most likely to mature as par, and those that inherited 2 L alleles have a much lower probability. And the same applies if you look at body condition and maturation in older males. So these would have already been to sea, except that they are in the common garden. And at 2 temperatures, the VGL3 effect was still there. So fish with 2 E alleles are the most likely to mature for a given body condition. And by body condition, I mean several months before actual maturation. So the next steps were to ask what links this variation in VGL3 to maturation? What are the physiological and behavioral steps that actually produce this massive effect on phenotype? And that was one project I pursued in a postdoc project with Tutku Aikanat at University of Helsinki. So we wanted to study if VGL3 is actually constraining how the salmon are using energy. And this brings me back to aerobic scope. So because aerobic scope is such an important concept for fish performance, we were wondering if VGL3 is constraining the ceiling or the floor of aerobic scope, so the MMR or the SMR. Now, by now, most people have switched to doing controlled crosses in these experiments because this allows us much better to select what kind of individuals we are comparing. So what we do is we use broodstocks that Luke has for making salmon releases in rivers. And then we just get some of our parental fish from those broodstocks and genotype them and select parents with specific genotype for mating. And then we produce families with known genotypes. And then we pit tag the offspring. So pit tagging gives us a unique ID to every individual. And then we can genotype those and select individuals for studies. But otherwise, this was similar to the first temperature experiment. So fish were tested for aerobic scope as juveniles. Only this was done at 11 degrees, so pretty cold temperature. We didn't find any difference whatsoever in SMR. Perhaps not surprising because it's a complex trait and it's only a one gene. There was a small but very significant effect on maximum metabolic rate, so the one that defines the oxygen supply capacity. So late maturing individuals seem to have a lower capacity for oxygen supply, and that means also their aerobic scope was significantly smaller. It's not a huge difference, but I wouldn't expect it from like such a complex trait anyway. It just made me think about the possible connection to thermal performance, seeing this result. But it also suggests that maybe VGL3 has some influence on energy budgets, and if the effect would be consistent across life stages especially, it could constrain maturation by this mechanism. So overall, I do not have time to go over all the studies that have used VGL3. There's a bunch of traits already associated with it. This is mostly from Craig Primer's lab and Tutku Aikanat's lab. But I wanted to highlight this new preprint with gene networks that are associated with each genotype, because it just shows that this single SNP can influence a lot of different physiological processes because it regulates other genes. So when other genes are turned on and off, then other pathways are turned on and off. And this study showed that a lot of processes in gonads are different between the genotypes. So from cell-cell signaling, transcription factors, which then turn on other genes, and cytokine receptors, etc. So it is possible that VGL3 is this sort of master switch or master gene. We still have a little bit of work to understand the whole pathway though. So to summarize things so far, salmon are vulnerable to heat stress. We know aerobic scope decreases, and we know VGL3 is linked to these physiological processes, including aerobic scope, but we still don't know, is VGL3 linked to thermal performance, and in general, are salmon life stages equally vulnerable to heat waves? So this is what the current project I'm working on is focused on. First, a little step back to thinking about why is it important that we address variation across life stages. A lot of people are interested in, are fish vulnerable to climate change, and do they have enough buffer in their thermal safety margin or buffer in their physiology to cope with an increasing temperature? And there was this big meta-analysis that compiled more than 600 species data and suggested that there is a big difference between life stages. So if they were just looking at adults and climate warming scenarios, there was no change in the thermal safety margin, so number of species that exceed the margin. But if they look at a sensitive life stage, then there's a much bigger proportion of species that are at risk. There was one problem, however, that a lot of the data was inferred. It wasn't tested physiologically in the laboratory. So we think it's important to validate these results also with comparable data across life stages. And this is what a postdoc who's working with me, Zara Cohen, is now studying. She wants to get thermal performance data across the life cycle of salmon, and they should be all acclimated to a similar temperature and measured using similar methodology. We are using the so-called CT max test, which is a relatively fast and standard way of measuring tolerance to acute warming. It doesn't necessarily reflect the effect of slow warming, but it gives you one aspect of their thermal performance. So basically the test is heating water at relatively fast rate, and then at the point where the fish can no longer sustain swimming-like position, they will start swimming sideways. That's the critical thermal maximum, and that's for the juveniles or bigger fish. Now for the embryos, because they are still in their eggs and we can't see what position they're in, this is based on when they stop moving. So we are filming the embryos during the test and then determining last movement. For the larvae, it's based on when they stop responding to touch. So anyway, it's a similar metric addressing when movement is hampered. So I'll show you all the data we have collected this year. This is reaching from embryo to smolt stage. So smolts would be 1-year-old fish that have prepared to migrate to the sea. We studied them in the spring when they were in this kind of intermediate phase. So first, from embryo to larvae or alevin stage, there is a clear increase in CT max, as would be predicted based on the theory that embryos are more vulnerable. But overall, the value is really high, considering that salmon are expected to be super sensitive to warming. And then we see a small increase from larvae to fry, but unfortunately, I think this is caused by a heat wave, because during our acclimations, we had an accidental heat wave because it was spring and the water was warming really quickly. So I think these fish got a little primer for the CT max before, and therefore their CT max is higher. But nevertheless, it's in the similar range as the larvae one and very stable across these life stages. So it doesn't look like a huge amount of variation. You may also notice that there's different sexes in the graph, and the way we determined the sex was collecting fin clips from all these early life stage individuals and genotyping them for a sex marker and VGLL3. So I can show you some results for VGLL3. It's pretty conclusive in that there is absolutely no effect whatsoever, and probably doesn't need a statistical analysis either. But I think for salmon this is great news. If there was a genetic link between CT max and life history, then perhaps heat waves could, in their own way, disrupt life history variation in salmon populations, but it doesn't look like that. So what else does CT max tell us in general? People have shown it's heritable trait, so it can evolve. It's probably caused by oxygen supply in the brain being insufficient at some point when the fish rolls over. So it tells us about neuronal function during warming, but it doesn't tell us about other processes that can be sensitive, but they just don't show up in the fast test. However, I like to compare these results to what's actually happening in the rivers. And there's a cool study from Canada where they studied behavioral thermoregulation in juvenile salmon. So comparing our CT max, 28 degrees, and other studies which have usually been like 28 to 31 at most, it seems behavioral thermoregulation begins a couple of degrees below CT max. So this study was comparing when during summer the river temperature increases, like determining the point at which the salmon will leave their territory in the flowing water and go into a thermal refuge. And if you see here, the things here in the bottom are actually little fish. So they are aggregating in this deeper area with colder water, and this is really unusual for salmon, like they do not want to do this or leave their territory. So perhaps they were already stressed almost to their maximum limit when this behavior started. So the onset of these aggregations is usually around 26 degrees, but it decreases the longer the heat waves are, which makes sense because the fish become more stressed. But in general, maybe CT max is a useful indicator of, for sure, behavioral thermoregulation begins below this temperature, like maybe a couple of degrees below this temperature. Oh, one thing I want to mention is that by collecting these data from different populations, or even if we do that and seeing there's not much variation, we could use this sort of metric in restorations and try to develop territories that are thermally suitable for juveniles that have enough thermal refugia for them. Now, I would love to show you some other aspects of the thermal performance between the genotypes, but unfortunately this experiment finished 2 weeks ago, so I really can't. I just want to show you how happy we were after it was done, 2 months of work, and this cute salmon in their little chamber. But maybe a few words about future directions still. So I didn't mention this, but VGLL3 is not the only large effect locus in salmon, and by locus I mean gene or genomic region or part of the DNA. In the same study that it was found, they found another one called 6.6, and that was even more significantly adaptive and important for adaptation, but it wasn't sort of related to age at maturity after population structure was corrected for, so it's been more in the background, and we don't really know exactly how and why this 6.6 is affecting salmon. But still, we've done a couple of studies, and other people have started monitoring 6.6 frequencies as well. And I want to show you something very complicated and a bit crazy, but bear with me. In one study, we Actually, in the same fish that we measured the maximum metabolic rate from, we collected their heart tissue. And the heart obviously pumps blood and is important for survival. So mitochondria in the heart are what produce the energy for the heart to function. And there is this enzyme that can be used as a proxy of mitochondrial abundance, so that in high throughput we can screen fish for mitochondrial abundance in the heart. So we did this across the genotypes in 2 different food treatments. And there were many interactions that are difficult to grasp, but for example here, VGLL3 late maturation genotype, so they have 2 L alleles. They had a significant difference between 6-6 genotypes. So 6-6L, VGLL3L has lower mitochondrial abundance. You might also recall the late maturation allele related to lower aerobic scope. In another study that was then completely different, it's using scale archives on wild salmon to look at their maturation in a river in Norway, and they find that in older data, there is a significant 6-6 effect, again in the VGLL3 LL in age at maturity, in males, specifically here, but in more modern samples from 2013-16, there is like an overall less effect of VGLL3 and less effect of 6-6. So it's just different time, different environments, genetic effects are different. So it just shows you what complexity we're dealing with, but I guess it's the way forward anyway, to try to get down to what are maybe the more generalizable patterns. Also, maybe more logically, I'd like to see more studies doing this sort of combining genetic and physiological variation into more modeling the stock and modeling ecosystem change. We need to know more about the microclimate or microhabitats, so modeling within the territories of the salmon juveniles, for example, what the effect of environmental variation is on their performance. For example, growth that is important for fitness and survival. I would also love to see that we use our monitoring data sets on this, given that a lot of us have decades of data already collected. So I'll just finish with some take-home messages. Heat waves are definitely imposing a selection on salmon already today in some rivers and in the future in some other rivers. But we should pay attention, especially when trying to improve the habitat quality, that it is thermally suitable for salmon and also other diadromous fishes, I should say. We also conclude that VGLL3 is this master switch for maturation, so life history, but it doesn't, based on these results, control thermal tolerance, which is good. And perhaps provocatively, we need to be able to forecast and prepare for the effect of climate change, and especially also heat waves and other aspects. I want to thank my funding sources, obviously, and for ICES, and my colleague Laura Uusitalo and Dave Reed for encouraging me to come here today. And thanks everyone for listening.
Thank you, Jenni. Deep breath. Do we have some questions for Jenni? Does somebody want to start? And you'll have to really stick your hand up because I can't see. Yeah, we have one over here, please. Welcome.
Thank you. Thank you for a very nice presentation, Jenni. Covered a lot of ground. I'm going to try to remember the marker, VGLL3. Great. So it was a marker of age maturation, And then at one point in your talk, you mentioned migration strategy was also associated with it. I wondered if you could maybe speculate, comment about how heat stress in all these latitudinally arrayed populations might select for a different age of maturation or a different migration life history type. Thanks.
Thanks. Yes, so migration strategy was found in one study where I think it was sex-specific, so only in one sex VGLL3 was related to migration activity, so when they're going to the sea, but not in both sexes and so on. So heat waves effect on migration, I think it would depend on the effects on growth primarily, because the delay of migration is usually also caused by lower growth. So it could be that if heat waves are decreasing growth, they are delaying migration and therefore making the whole lifespan longer. But I think generally it's happened perhaps in most salmon rivers that temperature increase has increased growth and therefore made migration earlier. So there's like counteracting effects because it's not all heat waves, it's also the average temperature is increasing. Did that answer your question?
Yeah, I've just wondered, so what is the marker anyway, the VGLL3, is that right? Well, in Ireland, we, what are called multi-sea winter fish, or fish around kind of 4 kilos or 5 kilos. We don't have any, well, there's a couple kind of reported every year and it makes big news, sort of 10 kilo fish. So I suppose there's anybody, and it's probably a much more kind of translating from the Baltic to the Atlantic is going to be a very hard thing to do anyway, but I suppose there's anybody maybe looked at Irish genetic data, and is there any lessons to be, or is, what kind of comparisons are there to be made?
Irish data, I'm not aware of anything yet, but I may have missed it, but I know there is a project ongoing at the University of Helsinki that is looking at Irish scale archives as well. So we'll definitely hear what is VGLL3 frequency in the different alleles there. But it's true that in much of the range, it's now more one-sea winter than multi-sea winter, for sure. But that doesn't mean that there still isn't this late maturation allele present.
Thank you. That was a really great talk, and there's a lot to take in and process still, but I wanted to go back to Dave's earlier question about migration strategy, and you talked about migration out to sea. I work more on adult salmon and marine influences on their experience at sea, and so I'm curious, your work obviously focuses on the genetic component of maturation. I'm curious if you've thought a bit about the environmental influences on that as well. There has been some work looking at declining growth with warming at sea, or in the adults, and then influences of that on maturation, and slower growth would cause them to come back later. So I'm curious if you think there would be carryover effects of that to the early life stages in the rivers that might influence how we should think about some of the work that you showed us today.
Like carryover from delayed maturation at sea to their river
their
performance.
right.
Yeah. Yeah, well, at least if they delay maturation, they will be bigger because the whole year of growth is still a lot. And then the offspring, so they will have a better chances in their start of life because eggs are usually bigger, and like this study found higher numbers of surviving offspring from the later maturing ones. So it could perhaps, let's say, compensate for the delay. So instead of having fish come in earlier, reproducing, some of them going back to sea, reproducing again, so on. We might have fewer fish that come in later, but they just produce so much offspring that the effect is sort of okay. But I guess this is the speculation. I don't know what other carryover effects there might be. Like, I know that genetically there can be differences in if L allele parents reproduced and E allele parents reproduce in the offspring survival, but if it's purely environmental, so it's just an environmental effect on growth, then I'm not sure if that has some.
Thanks very much for a brilliant talk. It was quite inspiring to see the amount of work that you've put into all that research. I was just briefly going to ask, we've had a similar situation with whelks on the English, in the Thames estuary on its north coast, which also is a boreal-arctic species. And it suffered a mass mortality during, well, sort of post-marine heat wave in 2022. And it seems to have a quite similar temperature stress threshold as salmon. And one of the things that we were curious to find out was whether there's a link between the temperature and the duration of the temperature stress. Because we've had subsequent years where those sort of same temperatures were reached, but not sustained. And we've not had the same level of concern. So that in one part of whether you've looked at sort of, I suppose, that acclimatization, new experiments, and then how long you stress them for, to try and then link that back to potentially, at which point do climate change scenarios suggest these rivers are going to be at risk of losing salmon? I don't know if you could maybe just comment on looking at that sort of temperature, duration, stress, and looking at future climate predictions of when rivers might be at stress. Thanks.
Yeah, that is a really relevant point. It's like with temperature, like with any substance, that dose is what kills you. Like, I'm sure that given if we used 20 degrees for long enough time, it might become stressful. If we use 25 degrees for one day, it might be too stressful. So it's all about the duration and also the previous history of the fish. Like often we see a rapid increase in temperature in the spring, and that's when the fish are completely unaware, like they've spent the winter in 0 degrees or 2 degrees, and then suddenly get 20. So I'm sure this is massively more stressful than having 20 degrees after 1 month of slow warming. So it is, I'm not sure if anyone is such a good modeler that they could do these predictions differently for these scenarios, but yeah, it would be cool to try. So far, because we need so many sample size, such a big sample size just to see the genetic effect, that we can't do all these experiments at the same time.
Thanks very much. Thanks for a wonderful talk, and thanks for a talk on salmon.
You're welcome.
In the UK, our most recent national sign of the effects of climate warming on the salmon has actually been in the winter on the juveniles. And we had a kind of almost a national decline in juvenile production a few years back, which was related to high winter flows and high temperatures. So cheekily, I've got 2 questions here. But my first question was, are you looking at any effects in the winter? And then the second one, which is completely different, is have you looked at, or has anybody looked at the potential effects of hatchery rearing practices on the distribution of these genes and these alternate genes? Thank you very much.
Thank you. I haven't planned on winter studies yet, but I'm definitely interested in that too. Like, I didn't know about that mortality thing in UK, so I'd like to talk about that later. And I guess, yeah, it's an interesting equation, because you would think that more suitable winter temperature makes them just grow in the winter, but maybe there's not enough food, or I'm not sure. And for the second question, hatchery rearing practices, yeah, they could affect it, but so far they seem to have maintained it. Like, maybe maintain is the wrong word, because we don't know what the ancestral distribution has been in most cases, but they have maintained some diversity. So because they don't look at it at all, and they just maximize the number of families from different parents, then it happens that in at least Finland we have good diversity in the stocked rivers for these genes.
Okay, I think you deserve coffee. If we have no more questions, I'm not
Thank
missing anybody, shout out. Then Jenni, I just have one thing for you.
Oh, sure.
Thank you.